Good afternoon. I would like to welcome you to our session, The Future of Life, Synthetic Living Cells. This session will get right to the heart of what it means to be alive and will provide insight into the latest research efforts to construct or reconstruct at least components of living cells. And perhaps by the end of the session, we will find out whether our scientists think it will one day be possible to construct a fully synthetic living cell. My name is Maria Siomos, and I will be moderating this session together with my colleague Yannis Salo from the European Research Council. We have 3 speakers with us here today, all of whom have received prestigious research funding from the European Research Council. The first speaker is Dr. Hannes Mutschler, and I'll just tell you a little bit about him. He studied biophysics at the Humboldt University in Berlin, followed by a PhD at the Max Planck Institute of Medical Research in Heidelberg, Germany. He then undertook postdoctoral training at the MRC Laboratory of Molecular Biology in Cambridge in the UK, and he is now an independent research group leader at the Max Planck Institute of Biochemistry in Martinsried, Germany. Hannes, please.
Thanks a lot, Maria, for this nice introduction, and thanks a lot also for organizing this Open Science Forum for our very specific and fascinating field of research. Of course, I would have loved to come to Trieste in person, but I think it will also be very fascinating like that. And so my presentation will be about our work on genetic systems and the possibility of creating systems that can synthesize their own biomacromolecules as a module for artificial or synthetic cells, however you want to call them. But I was also asked to give a brief introduction into the field in general, including some of the main questions that keep us up at night. And I would like to start with a very common concept, which most of you know intuitively, but it wasn't very clear until the 19th century that all known life on Earth comes from pre-existing life. Louis Pasteur was the one who figured it out in the 19th century, about 1860. He shaped this very famous quote, omne vivum ex vivo. And it essentially means that, for example, if you wanted to grow an apple tree, what you need is a seed. And for a seed, you need an apple, and then again, you need an apple tree. So life is a recursive system where you need already pre-existing life to make new one. The same is also true, of course, for animals. To hatch a chicken, you need eggs, and to have fertile eggs, you need a rooster and a chicken. And this is also true for the very fundamental, at a very fundamental level of modern life, which we will talk about later a little bit, is that also cells can only derive from already existing cells. So they don't assemble de novo from non-living matter, which is what people thought in the beginning, that life could just emerge spontaneously over and over again. Now, this, of course, raises a number of very important questions. Just the most obvious ones I will briefly discuss here as part of this introduction. The first one is, of course, how did life start in the first place? So this brings us to the famous chicken-egg paradox, where we know that in order to have a chicken, you need first an egg, and to have an egg, you need a chicken. And the question is, which one came first? Of course, this paradox cannot be resolved this way. Scientifically, what people have been looking into is the origin of life since about 50 to 60 years. People are doing this kind of research. So what you see here is a very nice depiction of the tree of life in relation to the presumed age of life. So these days we have bacteria, archaea, eukaryotes, including us humans here over at the side of the mammals. And what we know from biochemistry, molecular biology, and in general from evolutionary studies, is that all life derived from one primordial pre-organism or protocell, if you wish. So we all share the same biochemistry with some variations, of course, but the fundamental principles are the same. We all have the same molecular biology. And in fact, all life we know on Earth is related to each other. Now, given that life can only emerge from pre-existing life, of course, people are wondering how can life then at some point have emerged from non-living matter. We call this process chemical evolution. So this is what some people are interested in. Others wonder whether we can, in general, if nature can't do it at the moment, maybe humans can do it. So can we, in general, put or stitch back together living systems from non-living matter? So in this non-vegetarian example here, the question is whether we can reassemble, for example, a chicken. I like examples with chicken for some reason, whether we can reassemble chicken from its meat or non-living components. Let's put it that way. Then another question is, if we now switch examples and go towards something else that is very familiar to us, and that is cars. So cars were developed since the early 20th century and have undergone multiple steps of improvement and technological advances. And if we take this now as an example for a living system, it's now very sophisticated, but some people are wondering what could be the essence of such a car. Can we reduce it back to its main components, which is wheels, maybe an engine and a steering wheel? So can we strip it of its non-essential features? And that's also what some people try to do with living systems. So can we find the most minimal form of a living system? And because this is a bioengineering inspired approach where you start with a living system, you can also ask the question whether we can modify life to an extent that we can create life with new features that for example, produces natural products which are otherwise inaccessible by synthesis, for example, by chemistry. So this is more the application-based scenario. And then other people might ask, well, we all know that life is based on a common biochemistry. So the question remains on whether we can create a new kind of living that has a completely different biochemistry, so artificial biochemistry, if you wish, or maybe also some non-biological features such as electronics. So the fantasy can be quite. There's an open end of opportunities of how you maybe can create life, or is it only possible to create the life the way we know it on Earth? And finally, one of the big questions we have: what is the definition of life itself? So what is life anyways? So people are discussing this since also several decades, and there's more than 100 published definitions of the concept of life. And whenever you look into one of them, you find a loophole. So you can find an example of something that is not alive, so that doesn't meet the definition of being alive according to that definition. But so it seems to be very difficult to define life itself. So when you look into a normal dictionary, you very often find life is a list of properties that we connect to a certain entity, such as growth, the ability to grow, to metabolize, to respond to stimuli, to adapt and reproduce. So it's a list of properties, but you can always find examples of things that are alive and have some, not all of these properties. Then NASA, for example, has a very compressed definition of life that it's a working definition that is supposed to help NASA to potentially identify life somewhere else and not on Earth. So here the essence of a workshop was that life could be a self-sustaining system, chemical system capable of Darwinian evolution. And this already includes a lot of extra properties connected to that, because, for example, to undergo Darwinian evolution, you need the ability to replicate, to allow for variation, and to have natural selection to act on your entity. And a more fuzzy term is what is self-sustaining. So you can have long discussions about the fact of self-sustaining properties or systems. And there's many more definitions. Some are more related to non-equilibrium, thermodynamics, and information as a concept. And of course, there's not a time to go into most of them, but there's also some people that say maybe it's impossible to define life itself, because we have a gradient of aliveness that scales with complexity, for example. So a virus could have some properties that would give it a certain degree of aliveness, but it's also a very low complexity biosystem, and other things such as unicellular organisms that have a much higher complexity are also then more alive than viruses, for example. So there's different opinions on that, and this is what we also hope to a certain degree be able to answer at some point, or at least clarify with our research. But we're also experimentalists and we are pragmatic. And as I told you before, we only have one biochemistry on Earth. And we also have, that means that all life is related to each other. And what we know about all living systems is that they're all based on cells. So independently from whether you look at bacteria, but also at more complex organisms such as plants or animals, the basic unit or building block of all living things we know on Earth are cells. And then that allows us to rephrase some of the questions. So if we go away from the concept of life itself, then we can ask the questions whether we can build life cells from scratch rather than living systems. Can we understand how cells might have emerged or can emerge from chemistry, for example, on an early Earth? Can we synthesize a minimal cell from non-living components, and can we maybe create cells based on a different biochemistry? Now, if we look at least modern cells, the ones we know, we can have some basic characteristics that we can connect to or that we can associate with these cells. So what we know is that all cells that we know have a genetic system, for example, so they have the ability to store genetic information, to replicate it, and to pass it on to their daughter cells. In this case, then all cells we know of have lipid compartments. So they have a lipid membrane. That's something you will learn more about later in the next presentation. All cells have a metabolism, so they're able to harness nutrients from outside and build new molecules that is required to keep the system up and running, or they can also destroy certain compounds and use the energy that's stored in them. And all of that is based on catalysts, mostly proteins, but also ribonucleic acids, RNA. Proteins can also act as scaffolds and have different properties, also something you will learn later. And also another important aspect is signaling, so the ability to respond to your environment, because otherwise you're very disconnected and you won't survive for very long. Now, this is just the core principles of a cell. Of course, in reality, modern cells are much more complex. So if you look at this animal cell, you have many different subcompartments, you have thousands of proteins, you have very long DNAs, metabolism is utterly complex. And of course, it would be very challenging to build such a system in the first place, and maybe even impossible now. So what we're trying to do is build much simpler systems at first. And there's different approaches of how you could build such a minimal or proto or synthetic cell, depending on the angle you come from. So there's the famous top-down approach, where you already start, as you've seen in my car example, with a living cell. You try to reduce it in a way by keeping the cell alive and then reduce it down to its most minimal set of genes that are required to keep it alive. And most famous for that is the Craig Venter lab, or Craig Venter, whose lab managed to build this not build, but to reduce an existing genome down to a level where you can make a synthetic bacterium that can survive with only about 470 genes, which is quite impressive. For us people that do the bottom-up approach, this is still fairly complex. So here in this approach, we try to build systems that are alive to a certain extent, or maybe have at least life-like properties from non-living components. So we look at life as nature as a toolbox, and use proteins and nucleic acids and lipids and so on, and then try to stitch them back together. And of course, one of the major goals is to build something that resembles a living cell. And as said before, there's the origin of life approach, where we try to see how can something like that happen in the first place. So how could the first protocell have emerged under prebiotically plausible conditions? But this is a historic question, so we will never be able to really tell how it exactly happened, but we might be able to understand how it can happen. Okay, in particular, the bottom-up approach has is very flourishing at the moment. So there's different national and international networks that aim to build such a synthetic cell from the bottom up. As you will hear later about BASIC and from the Netherlands. Then there's Build a Cell. This is based in the US, and Maxim Bio based in Germany. And we all are interested in this bottom up approach, using different methods and also slightly different interests sometimes. So after this introduction, I would like to briefly come to what we are doing as part of our research. So we are interested in whether we can reconstitute one of the core features of modern life, that is the central dogma, which most of you might know from textbooks. So this is the flow of information in living cells, which is based on DNA replication, transcription of RNA as a transient template for the synthesis of proteins. And this is at the core of every living cell we know. And the question is whether we can build something like that in a way that it can keep on running for several generations. This is what we're trying using a bottom-up approach. We're also looking into systems that are simpler and can act as a model for the origin of life. So, for example, systems that work without DNA. We're also interested in whether we can combine both self-replicating RNAs and DNAs in a combined bottom-up and top-down approach to see if we can build hybrid cells that have both a replicating RNA genome and a DNA genome. And also, last but not least, we're interested in protein-free systems, again, relation to the origin of life question, because to make proteins, you need very complex machinery. And some people wonder whether life might have started with a much simpler system where there were no proteins, but only nucleic acids. And for the sake of time, I will briefly talk about this, our work on the minimal central dogma, and later about these two systems very briefly. Okay, so can we build a minimal central dogma starting from macromolecular building blocks that are already provided by nature? And in order for this to happen, what you need to assemble at least 150 genes together the right way that they can carry out this activity. So this is not a trivial, because if you zoom in one layer in terms of complexity, so this is still not a full picture, what you see is that you need a lot of reactions to happen in a concerted manner at the same time in a way that you have the replication, for example, of DNA. You need a so-called RNA metabolism where you make precursors that are required to build the so-called translation machinery, ribosomes, I'm sure most of you might know, tRNAs, translation factors, and so on. And they would feed back into the system, make all the proteins again required to carry out the different activities, such as DNA replication, and so on. So it's a recursive system again, where each reaction serves to keep the network itself up and running. And you might wonder whether this small number of genes is actually sufficient to rebuild a minimal cell. So when you look into nature, it might be possible indeed. So maybe not a free living cell, but maybe a cell that can survive in a very specific environment. So when you, for example, look at this very minimal cell, it's an endosymbiontic insect called Nasuia deltocephalinicola. It lives in these leaf hoppers as an endosymbiont and produces certain amino acids. And this is what these cells look like. Not very well shaped, but they are alive in the sense that they have a genome, a very small one, only 2% of the E. coli genome. But what makes them so interesting for us is that they encode for the entire minimal central dogma. So they have DNA replication, transcription, and protein synthesis. And this is what we find very interesting, because it tells you that in principle you can build a cell from that if you live in a very specific environment. So it doesn't have a proper metabolism. It steals most of the components from this leaf hopper. But in a way, it tells us that it might be possible to build a minimal cell, but this cell will not be able to survive outside of your test tube, and then again, you can see as a safety net, if you wish. But we would be already quite happy with having such a basic thing up and running. So what you would need for that is you need to encode and replicate the required genes on a DNA genome. So for the minimal central dogma, you need to also regenerate the proteins and the genes you have encoded on your genome. And only if you can do it, you can have at least partially self-sustained replication. And then what you can do next is put this system to compartments. We are not very good at that, so other people are much better at that. And then you can try to couple this to growth and division, make it more and more cell-like, and then add features maybe at some point, such as a proper metabolism, which allows the system to become more independent. And what we looked into first was in the aspect of DNA replication. So we're not the only ones doing that. There's also other people from other networks that do that. And we were wondering if we could at least build this subnetwork here, the DNA replication part, where we make a polymerase that then replicates DNA. And because we don't have yet the ability to regenerate protein synthesis, what we do is we add so-called in vitro translation systems. One of them is called a so-called pure system. It uses recombinant elements and it's very clean, which of course helps us in bottom-up research because we know what's in there, so it's not very messy. And you can make it by yourself, or we can also buy it, but then it's hard to customize. And we use these in vitro translation systems to boot up our systems. And at some point, of course, we hope that we can also encode them on our genome. And one project we recently started to finish in a way that it allows us to start a new stuff is the ability to replicate DNA coupled to in vitro translation. So it's not only us, but also other groups show that you can couple in vitro translation to DNA replication. So you make your replication system based on proteins in one reaction, where then this polymerase you make starts to replicate DNA. And we looked into the challenge whether we can replicate something that is of the size of a Nasuya genome. And in fact, this was possible. However, we cut our genome into small pieces called plasmids, so small circular DNAs, where you can add or leave out genes. So it's a kind of a modular approach. So it is possible to now replicate genomes that could, at least in principle, encode all the information necessary for a minimal cell. However, it's currently very unregulated, so it's more like a virus. It makes as much of its DNA as it can, and this is something we need to work on. And also what is very tough is that you indeed regenerate all the components that are encoded on this DNA using the in vitro translation approach. But so what we do is we look into this using mass spectrometry, whether we can make the components of the in vitro translation system, which are also components of the minimal central dogma. And so far we are one order of magnitude away of fully regenerating these proteins. So there's still a lot of work to be done. In particular, one of the main bottlenecks is the efficiency of these in vitro translation systems, but there's a lot of room of improvement of how you set up the reactions. So we're optimistic that at some point we might increase this number efficiently. But we believe that's maybe semi-autonomous replicating systems that make at least some of their components themselves is already something that might be possible. And very briefly, I have to check the time. I forgot to start my timer. So I hope we still have like 2 or 3 minutes. What we're also interested in is these DNA-free systems and hybrid cells. And here what we try to achieve is to have systems that are independent from DNA. So we use replication machineries coming from so-called bacteriophages, so viruses that can only infect bacteria, so they're totally harmless for humans. And we steal their replication machineries and try to build RNA genomes where these replicases here can then use a RNA genome to make messenger RNA, which then in a in vitro translation system, for example, can be produced in your, so this means gene of interest, into a protein that has an activity. And so this is just one example where we used components from this MS2 phage to make proteins from an RNA genome. And here you make a fluorescent protein, so you can easily watch the formation, or you can make enzymes that turn over a small molecule, and then you can follow the activity. And here what we try to achieve, in fact, is that we have self-replicate or replicable mRNAs, where we completely independently from DNA can replicate those messenger RNAs. We call them ribogenes. And just an example, this is in vitro work where we try to make RNAs that, for example, encode for antibiotic resistant genes and then replicate them. And then if you put them into E. coli, the hope is that they can then help this E. coli bacteria to survive under selective pressure just based on the replication of RNA. And this requires a lot of optimization, just very briefly. So when you replicate RNA, in particular long RNAs, what happens is a very frequent event for the replication of RNAs that you have replication or the formation of parasitic RNAs, so shorter RNAs that replicate much more efficiently than your good RNA, and then they poison the system. And that's maybe also one of the reasons why nature chose DNA in the end, amongst other reasons. But what you can do is you can optimize your replication machineries, just as an example here, where we use different cofactors to improve the ability of the system to replicate long RNAs without the formation of parasites. And here what we essentially want to achieve at some point is that we start from a wild-type bacterium where we have a DNA genome, and then we hope that we can transplant genetic information from DNA onto our ribogenes so that they can then complement the missing genes that have been deleted on the DNA. And this opens many different interesting aspects. But to achieve that, we need to develop different technologies or alter existing technologies to allow this to use as a toolbox to create those synthetic cells. And then you can target different genes. You can look for questions as whether you can transplant very important core genes from E. coli onto ribogenes, or maybe also artificial metabolic networks or synthesis pathways, which I didn't write here in this text box. So there's a lot of things you can then do, and the questions we hope to answer is whether we can build cells that resemble ancient cells that maybe at some point came as a transition from an RNA world to a DNA world, where some people speculated we might have had hybrids at some point. But the question is whether modern cells can survive with partial RNA genomes. This is also very interesting for us, because if it was so, then you could use these two different genomes. One is replicating with a lot of errors, so it allows you to do a lot of evolution experiments, whereas the other one based on DNA is very has a high fidelity during replication, so it evolves or mutates only very slowly. And the question is whether you can leverage two differently evolving genes for biotechnological purposes, such as the evolution of metabolic networks to make some natural products. And I hope I'm in time. I'm here at the end. I would like to thank my team who's working on all of these different projects, my collaboration partners, the main collaboration partners here, and of course, funding, in particular the ERC for the hybrid cell project I just showed you. And of course, I'd like to thank you for your attention. Me?
Thank you very much, Hannes. We can now move on to the second speaker of this session, that is Professor Ulrich Kaiser. He received his PhD in experimental physics from the Leibniz University of Hanover in Germany. He undertook postdoctoral work at the Kavli Institute of Nanoscience, which is now in the Department of Bionanoscience at the Delft University of Technology in the Netherlands, and also at the University of Leipzig. He then joined the University of Cambridge, where he is currently a professor of applied physics at the Cavendish Laboratory in Cambridge.
Thank you very much, Maria, for this kind introduction and the possibility to show our work here on controlling flow through membranes. And specifically, I want to talk about how we can actually build synthetic membrane proteins using DNA. And I would like to pick up a little bit from what Hannes just explained. And he has this very nice slide where he summarized the main properties of cells. And one of the most interesting part for me of this is that you always see this many compartments in these kind of cells where you have the nucleus and the other cellular organelles. And he also broke this down into this very nice slide picture where you have this compartment that is surrounding a cell, and then this minimal system that actually runs. And there's a metabolism happening inside with catalysts that are protein and RNA. There's a genetic system that encodes information of these kind of systems, and this is of course carried by DNA and RNA. And then there's one important thing that you also mentioned very briefly on this, and this is signaling, because of course what you need to do in this kind of system, if you have a compartment, yeah, and you know this from your home, if you want to live there, you normally acquire you need some food that you bring inside, and you also have some garbage then that you have to take out. So actually, the interesting part is that you actually, in this, if you have a compartment, you also need to, the compartment is not closed, but actually it has to allow flow through this kind of wall that separates you from the outside world, okay? So and I would to first quickly tell you a little bit about the compartment, and I think this is also a nice connection to the next talk, because there, Marilene Dochterom will show you how to make these artificial compartments in microfluidic systems. But fundamentally, what I want to just quickly tell you what these compartments are made of, and this is lipid membrane. So if you zoom in here, this is how it looks. It's a lipid. It's a lipid here. So these kind of small molecules are called lipids. They have a polar head group and an apolar hydrophobic carbon chain. And if you think about this on a very simple level, this is like soap molecules. So you have a hydrophilic head group and a hydrophobic oil-loving tail group. And if you put them together, they're actually forming these nice double layers that form a barrier for any substances that want to go in and out of your compartment in this kind of system. So the lipid double layer keeps molecules and ions inside the compartment, so you can confine things to run autonomously inside these cells. Again, it's a membrane, so it acts as a barrier to unwanted exchange of material with the outside world and keeps things nicely confined inside. But for the proper functioning, it's very important to control the transport in and out of the cells. And this is actually also called signaling. So this is a very broad thing. So you actually want to transport ions in or out of the cell. And this is actually happening when you have your nerve cells firing. You want to control the flow of ions through these membranes. And in these kind of artificial systems, of course, we also will have to have some kind of metabolism going on. So you will produce waste products, or you will use building blocks that you have to replenish through the systems. And what we are interested in my group is basically looking at these components and how we can build these kind of controlled systems to control the transport in and out of the cell. And of course, there you can learn a lot from what nature does already. So here you see a picture of a cell membrane. Here again, you see these lipid molecules that are forming this bilayer. And then actually what cells do, they use membrane proteins that are sitting in the cell membrane to actually regulate the transport. And cells, of course, evolved, like Hannes Mutschler said, to very complex and very different structures. So here's one of the biggest protein complex that you find in our mammalian cells. It's a nuclear pore complex. It has a size of around 150 nanometers across. It regulates the genetic transfer of information of RNA basically out of the nucleus and proteins into the nucleus in our cells for replication and replicating DNA. But then also there are these kind of smaller porins where you have a 5 nanometer size, so it's 30 times smaller, that is directly sitting in a lipid membrane here and allows the transport of sugar molecules in and out of bacteria. Okay, so you have a vastly different size scale in these kind of systems because all these systems transport different things. So this can transport large protein complexes, and this is small sugar molecules. So if you want to have any hope of replicating life, I believe we really need to be able to replicate these structures as well from scratch, from a building materials that we understand, okay? And I'm working at the Cavendish lab at the University of Cambridge, and of course we call this the home of DNA because the structure of DNA was resolved with based on the crystallography images of from Rosalind Franklin by James Watson and Francis Crick. And here you see this model here. And of course, the important thing here that I want to just, because I really like the sentences, has not escaped their notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material, because what they actually understood is how you fit the DNA together into this beautiful double helix. And this is just a quick reminder for those of you who don't know this. So DNA will be used as a molecular building material, and we can do this because what Watson and Crick understood is that the molecule of life, DNA, has 4 letters. It's A for adenine, T for thymine, G for guanine, and C for cytosine. And the nice thing, what they understood is that actually A always binds to T and G always binds to C. So if you now look at this molecule, you could make this molecule, this polymeric of DNA. So you can make one DNA molecule with a sequence A, T, G, G, T, C, and you make another one with a sequence T, A, C, C, A, G, and they will always fit together in this kind of way with a Watson-Crick base pairing mechanism. And this mechanism actually leads to this beautiful double helix. This is a photograph of the model Watson and Crick made back in the day that you can see in the Cavendish lab. And fundamentally, if you now look at the length scale of these systems, so these multiporin pores are roughly 5 nanometers across, and the DNA has a diameter of 2 nanometers. So just as a reminder, this is a 0.000000002 meters, so it's pretty small, everything. So we really need to have a very high control over the molecules to actually build anything that can be useful, okay? So what we want to now do is, because we understand the base pairing mechanism of the DNA, we believe we can now use the DNA to build whatever we want. Yeah? And I want to just quickly show you how this works. So again, this is a double helix. So this is a fundamental structure we will deal with. So you see here the base pairs. And again, I think the right picture in your head should be that we actually have these small DNA molecules that we can program with our letters. And then these can be seen like Lego building blocks that fit together in a certain way, because again, with a sequence, we can program how they assemble. And what people then figured out in the 80s, so around 40 years ago, is that if you make 4 DNA molecules with the right sequence, so a blue one, a yellow one, a red one, and a green one, you heat the whole thing up, mix them together, cool them down, you actually can make a 4-way junction out of DNA. So this is DNA molecules, and you can make a 4-way junction. Again, the diameter here is 2 nanometers, so this is exactly the size of one of our membrane proteins that we saw before. So, and the nice thing about these Lego bricks is that it's like if you take a Lego set at home to your child, and then you don't have to spend hours assembling it, but all you do, you shake the pack, and after shaking the package, you open it and it's assembled, and this is exactly what we can do with a self-assembly of DNA in this way. So what we can do now, we can actually also attach any chemical group to these DNA molecules. We mix everything together. We heat the mixture up. So it's almost like molecular cooking, and then you cool it down, and you get billions and billions of identical structures that are perfectly self-assembled into the right structure. Okay, so and there's a famous example, because you actually can create happy DNA. Basically, this is famous smiley face that Paul Rosamund published on Nature in 2006 that basically inspired the whole field that we are doing here, because you can actually force a DNA molecule into the shape, so you can really make any shapes that you want. And what we also know already from the things what we saw before about life, that of course structure is very important. So you need to be able to organize molecules in a certain way. And here you see a zoom in of the thing. So the idea is here that you see the single strand DNA molecules with the different colors. You see the double helices, and here are the 4-way junctions that I showed you before. So basically, the idea here is to say that DNA is not only carrying genetic information, but since we understand how to encode the information, we can also use it as a molecular building material. And just as a reminder from length scales, the smiley face is around 50 nanometers. So one of these holes is 5 nanometers. It's exactly the size of one of our protein membranes that we want to build with this system. So I now will show you just quickly 2 examples what we did in my lab to build these kind of different structures. So we have an inspiration from nature. This was a nuclear pore complex that has a size of around 150 nanometers. So the question we asked now ourselves, can we actually use our DNA nanotechnology to actually build a structure that approaches this size in this way? So this is a computer-generated image, of course. It's not the real structure, it's too small. I will show you a microscopy image in a moment. So each of these red bars here is a double-stranded DNA molecule. So basically what we do here, we mix around 200 of these strands together, and then it folds into this kind of structure. You also see here a rendering of the lipid membrane, and this guy is punching a hole in the lipid membrane with around a size of a couple of nanometers. So if you look at this just in a more simplified way, of course, DNA normally doesn't interact with lipid membrane, so we have to do something, but here we can just attach small cholesterol molecules. This is a molecule that you find, for instance, in butter. So it's likes oil. So, and again, the lipid film is like an oil film. So by attaching a lot of these cholesterol molecules, you can actually force the DNA to punch a hole in this lipid membrane. So if you see now, this is a funnel-like structure. So if you look at it from the top, it has a diameter of around 22 nanometers, and the inner diameter is 6 nanometers, not quite the size of the nuclear pore complex, but it's approaching this. Yeah, and if you want to see how this structure looks like under an atomic force microscope, here you see the structure, here you see the funnel. So this part will be going into the membrane, and this is the top part that actually is supposed to mimic the binding sites of the nuclear pore complex. So this is working, and the size here is 22 nanometer, and I think it's nice to see a video here. So this is a computer-generated simulation by our colleagues from Urbana-Champaign that I will run for you. So you see here, this is the double-strand DNA helices are shown in blue and yellow, and this video will run several times, so you can look at it a couple of times. Here you see the molecule, now it's relaxed. You see the lipid membrane, so it's already inserted in the lipid membrane. You see the cholesterol molecules. Now if we zoom in, the red balls are actually now ions that can flow now through the punched hole in this kind of lipid membrane. So we run the video again. So again, you see here the structure of the DNA molecule. Of course, a quick reminder on these kind of length scales, everything is a bit floppy. So you see the DNA is moving a little bit, and then at the same time, the red balls that are moving here are the ions that can now flow through the lipid membrane that this large channel attaches to. Okay, so you can really make an artificial ion channel, or this guy actually can also conduct proteins and other larger things, as other people have shown as well. So you can really make a structure inspired in this kind of way. And of course, the other thing that we probably need to do is, if you want to have a tighter control over what's going through, you also want to have a smaller molecule. So we tried then to make a smaller system inspired by a multiporins thing that only allows small molecules to go through. And then we made a much smaller structure. So again, you see our blue and yellow theme of the double-stranded DNA double helix. So you see the one strand is running through here, another is running through here. This structure is much smaller. So the other one was around 50 nanometers in length. This one is only 30 nanometers in length and consists of only 4 helices. The cholesterol molecules that actually anchor the pores in the lipid membrane are actually shown here in this red as red dots. And if you look at it, how it sits in the membrane, this is a computer-generated image. Again, where you nicely see the double helix running around. Here you see the cholesterol molecules, maybe here in yellow, and then you see the lipid molecules that are also, of course, disordered sitting in this membrane. Okay, so this whole structure is around 4 nanometers in size. So it's really approaching the molecular size of these kind of multiporin things. And what we can now do, we can actually measure the ionic current flowing through these small structures and compare it actually to the large structure. So here I brought you some data where you look at the ionic currents through the large and small DNA channels. And if you look here at the conductance as a function of time, when one of these things punches a hole into a lipid membrane, you see actually here, this has a conductance of 300 picosiemens, and this one is around 40 nanosiemens. So if you look at the histogram of these structures, you actually see that there's a difference of almost a factor of 100, yeah? And 2 orders of magnitude in structures, both in size and control over conductivity, is very important, because this is the way how you can maybe build a future system where one of these channels actually allows the transport of nutrients into the cell, and another one that actually controls the transport of ions in and out of an artificial cell. Okay, so you really want to have the possibility to build artificial membrane proteins, and I think with this kind of large, vast size difference, we can show that we can do that. And then there's another important property of these kind of systems, because of course what we also do is we somehow compete in this project with nature. So we want to actually outcompete or be better than nature in some of these properties. And the interesting part of this kind of systems is that they do something else as well. So this is a small structure that I showed you before with the cholesterol group here. This is shown in red here. And these small yellow or golden balls here are indicating lipids. Okay, so and one is highlighted here. And in this video, you see that the lipids don't stay at one side of the membrane, but they can actually switch sides of the lipids. So we have another way of controlling transport in a lipid membrane. We can transport molecules along here, but we can also see that a lipid can actually switch from one leaflet to the other. And this is normally a process that never happens in cells, or it shouldn't happen in cells, because cells use this lipid flipping to indicate that they are not feeling well. So dying cells very often lose this asymmetry in the lipid bilayer, so they have a lot of these scramlases, they are called, that can actually mix lipids molecules from up to down. Yeah, and our system here, this minimal ion channel, also actually allows the flipping of lipids. And the interesting part now, what you can do is in the simulations and also in experiments, you can now look at this. So again, the lipid head groups are now shown on one side of the leaflet are shown in purple, and the other side is shown in green. And then after, if you start the simulation here with our structure, and here you see the small ion channel, then you see that after around 2 microseconds, lipids from the lower leaflet appear in the upper leaflet. Yeah, so the lipids switch to the other side of the system. And now you can just count the number of crossings per microsecond. You find that you actually have 10 to the 7 lipids per second flipping here. And just to give you one headline figure where we now can, I think, outperform nature, because these our artificial DNA lipid scramlases work possibly 1,000 times faster than natural counterparts. So we can actually build now an enzyme from these DNA molecules that are much faster than the natural system. I will just quickly check the time to see. So I'm almost perfect in timing. So I think what I wanted to show you is that we have the capability now to build synthetic DNA proteins that can mimic essential functions from the natural counterparts. And what I showed you is that we can make DNA ion channels. We can also make larger DNA porins. We also looked at minimal ion channels that are just much smaller, just a single helix. And I think these systems might actually be a very interesting pathway for addressing this issue of how do you control transport in and out of these artificial cells that we will see in the next talk. So basically, I think DNA nanotechnology allows for building complex membrane proteins. And I think some of our synthetic DNA enzymes that we can build now outperform natural systems. And just one last question that I want to end here to think about making an artificial cell. So if DNA structures can actually perform these complex functions, not only for carrying information, but also signaling, maybe we can build an artificial cell from DNA and lipid components only, so we don't need proteins. Of course, this will be a challenge, but some people also, former colleague Kerstin Göpfrich in Heidelberg is working on this. And with this question at the end that I find exciting, I would just like the people who did the work and the funding, and thank you for your attention.
Thank you very much, Ulrich, for that very interesting presentation about how we can make pores to get things in and out of cells. Our third speaker now is Professor Marjolein Dachterom. She was initially trained as a theoretical physicist. She undertook graduate and postdoctoral training in Paris and also in Princeton and the Bell Labs in the USA. In 1997, she returned to the Netherlands as an experimental group leader at the Amolf Institute in Amsterdam. And in 2014, she moved to the Delft University of Technology, where she heads the Bio-Nanoscience Department, which is part of the Kavli Institute for Nanoscience. So, Marjolein, please.
Well, thank you very much, Maria, and also Janne and the ASOF and the ERC for organizing this session. It's a real pleasure to speak after these two previous very interesting talks. So what I would like to do is talk about actually a collaborative project that we started in the Netherlands, but also there are very comparable initiatives around Europe and the world, as Hannes already explained, about the bottom-up approach to building synthetic cells. So as was, I think, clear from the introduction that Hannes gave, even the simplest cells as we know them on Earth are still very complex and difficult to understand in detail if you compare them, for example, to a virus, which is relatively simple and already difficult to understand. And it's also therefore clear that any approach, all these approaches that now are developing to build bottom up from scratch these cells, or even to use a top-down approach, are really in need of collaborative projects. We can all think about individual components of cells and be really good at building those from scratch, but it's really about the integration of these different systems that in the end we need to do to get there. So I'm going to spend just as an example explaining the collaborative effort that we started, but I think it is a sort of something that we have to do, of course, also beyond national programs. But let me start, and while doing this, I will give a little bit of an example of the kind of research we do in my own lab, which contributes to these larger collaborative efforts. And let me therefore start by just showing sort of the background or some movies of what inspired or what inspires me to try to build synthetic cells. And that has to do with very fundamental questions. So what you see on the left is a movie that you may have seen before. It's a very interesting, let me start it again, or a pretty movie of what you might see if you were able to make yourself small enough to insert yourself in a living cell. And you see an artist impression. It's obviously not real data, this, but it's an artist impression of a motor protein that is carrying a cargo through, in this case, a large eukaryotic cell. And it's bringing material, building material from where it's produced to where it's needed. And this molecular motor is actually a good example of how much we've learned about the molecular components of cells in the last decades, starting in the 90s and since then. We've become very good at not only sequencing the DNA, the genetic information of all kinds of cells, but we do know quite a bit about individual molecules and what they do in cells. I think it's, however, still fair to say that if you ask, okay, how do all these individual molecules, of which we know a lot, how do they actually work together in time and space to make something like life, if you want to call it that, on a cellular level possible? We really don't understand much at all. So what I'm showing on the right is a picture from a Japanese lab of one example of a unicellular organism. So in this case, it's a fission yeast. It's a single cell organism that shows very beautifully what all cells are able to do, whether it's simple cells or complicated cells. And this is to reproduce itself. So what you see in the movie is single cells that have in green their genetic material, which needs to be duplicated, copied, and then the cell needs to grow exactly twice as big and divide, in this case, in the middle, and you go from one cell to two. It's the chicken. Well, no, it's not really. Maybe it's not the chicken and egg yet. It's a cell building a copy of itself. And what we as a consortium are really interested in figuring out is sort of what does it minimally take, or what molecular components need to work together in time and space, in which way, to make these fundamental processes that all cells are able to do possible. So it's growing, transmitting information, genetic information in the form of DNA, and then duplicating into two new cells that represent copies of themselves. Now this, for our consortium, is a very pragmatic definition, if you want, of life. We want something that reproduces itself. But as also already pointed out earlier, it's maybe not complete yet. So we are not asking it yet to communicate, to respond to signals, or to evolve, which you can all argue are very important aspects of real life as well. So the approach that we take, and was already explained, is to ask, okay, we know these individual components, and can we find a way to bring these together piece by piece and build a minimal cell-like system, if you like, that is able to do the things that I just explained. And we are starting from molecular components that are derived from biology as we know it around us today. So it's not about the origin of life. It's also, in our project at least, not about using alternative molecules to create life properties, but you can easily imagine how this can be integrated with what we just heard from Ulrich Kaiser, with non-lifelike molecules that can perform similar functions. Because we are really interested in the question of how do we need to put molecules together to get this biological function. And it's just a choice to start with biological components as we know them, but you could certainly do that otherwise and still get the same function, and maybe have even advantages in some respect by doing that. So even if you want to do the sort of the minimal bottom-up thing, there's still a lot to worry about. So we think that even in the most minimal system, you have to worry about metabolism, energy production, as was already introduced. I don't want to go through all of this, but you need to replicate DNA, somehow organize the chromosomes so that the DNA, when it's replicated, gets divided to 2 cells, worry about the container, the shape, that it's mechanically stable. So even for what you might call the simplest system, there's a lot to worry about. So why do we still think that now may be a time to at least try this, and that it's maybe feasible on, I don't know, 10, 15 years time scale to get to an integrated system like that? And that is what I already said. In principle, we do know the molecular parts. We know from simple cells what is the genetic code. And for example, this top-down approach presented by Greg Venter, where they stripped a simple cell down to its minimal components, gave us an idea of what is the order of the magnitude of molecular parts that are needed. And it was already presented for that particular publication. They came down to 473 components. So we don't know if we need all of them, whether we can have alternative sets of molecules. And this is the questions that we are after. So again, we know the molecular parts, but how does life or life-like properties emerge from these molecular interactions? What is minimally required? And maybe more importantly, what functions are minimally necessary? And this, of course, is a bit related to what you ask it to be. So we are looking for a system that we thought might be modular. So we need a system that is able to fuel the cell, to take care of metabolism, energy production, but also production of small molecules. Our system will need something to process DNA, to replicate it, but also to translate to proteins. And in the end, the container needs to be there, and it needs to be able to grow and divide. So that's also how we set up our collaboration. So we have different groups, and I'm just going to flash their faces, not that I will mention them all, but just to again emphasize, you really need biochemists in this case, who are experts in membrane proteins, and reconstituting, for example, ATP production with the help of membrane proteins in artificial cells. In this case, they are made from liposomes. And again, referring to the previous talk, in an alternative approach, you could maybe replace this with DNA origami-based structures. We need expertise on in vitro gene expression, translation, DNA replication and control. So the pure system that was already introduced, which is a controllable system, even though it's still very complex, that allows you to, in an artificial cell, produce proteins based on DNA that you feed to this system. And then there's the more mechanical side to things, is how to build a container that is stable, that can grow, that will not explode when it's trying to divide. And then a system that, or maybe multiple, that are responsible for spatial organization. Because I keep emphasizing, you can have all the molecules that are able to produce something, but at the end of the day, it's also important that especially DNA ends up in the right place and the right time. So you do need some kind of spatial temporal control of at least some subset of these molecules. So this brings me also to my own group, what we are interested in contributing, and this goes back to our interest in the cytoskeleton. So I'm just showing here now 3 versions of the same process, which is in this case examples of more complex eukaryotic cells. And one of the important things that also these cells have to do is to to faithfully separate copies of DNA or chromosomes. And certainly more complex cells like our cells, human cells, but all eukaryotic cells use a network of polymers called the cytoskeleton to build a machine called the mitotic spindle that is responsible for separating these chromosomes. And it's fairly textbook material in a sense. You may have seen it before, where this network of polymers fixes itself to the chromosome pairs and then separates them apart, such that exactly half of each set of chromosomes ends up in the left and the other half in the right cell. And this network of polymers, unlike our own skeleton, is actually a very dynamic system. It's able to constantly reinvent itself. So at some moment it can be a system to separate chromosomes. But then at another time of the cellular life, it's a highway system for motor proteins that I showed in the very first slide. So it's very dynamic, and I'm going to show again an artist impression movie that zooms in on really the molecular details of this one of these polymers, emphasizing again how dynamic they are. So each of these wires or polymers or cytoskeletal elements, if you want, is actually constantly being built and being taken apart again. And it's this constant switching between growing and shrinking that allows it to really adapt to the particular function that this polymer system is responsible for at any moment in time. So what we have been for many years interested in is, can we sort of bottom up, starting from these polymers, which you can isolate from cells and reconstitute in a cell-free system? Can we learn how the dynamic properties and also the force-generating properties of these polymers can really contribute to the spatial organization of cells? And to be able to do that, there's two technical things that I have to show. One is, how do we make our artificial cells? And it was already mentioned. You can use microfluidic techniques to make bubbles, if you wish, of a cell-like material in an oil-like environment. And this gives you a surrounding of the artificial cells that is just a monolayer of lipids. So it's not yet a membrane. And other groups are actually using similar techniques to make real membranes as a container. There's some advantages of using these monolayer microfluidic droplets, as they are called. And for us, they're very convenient cell size, at least containers that you can easily make on chips that we put under our microscope. And you see an example of that here. And then the content of these artificial cells, we also need to control. So we've, over the years, done many experiments showing that you can reconstitute the dynamic behavior of these individual filaments faithfully. So I'm showing just fairly quickly a series of pictures where on the top you see a real cell where the function that we're trying to reconstitute is shown, and on the bottom our version of it, which is completely stripped from the cellular environment. So individual polymers can be reconstituted. They are dynamic under the microscope. We can also study the force-generating properties. So whenever these filaments hit a cell boundary or a chromosome, actually simply by growing and shrinking, they can generate forces, which we are able to measure. And then we finally put these polymer systems in our artificial cells and control the nucleating properties, and thereby can really study the self-organization principles of these polymer systems in a cell-like container that is much simpler than a cell, and that allows us really to understand in a lot of detail why the polymers organize themselves the way they do. And as is very important for real cells, is that these polymers need to be controlled so that their dynamic properties are at any moment adapted to the function that these polymers have. And this is also something that we can now reconstitute in our artificial cells. So this is, I think, an example of the kind of bottom-up, step-by-step building up you can do to study a specific biological function. In this example, it's about understanding the force balance, the dynamic properties of this polymer system. How can it organize itself in a way that is useful when a cell is trying to divide its chromosomes? Now, of course, if you compare the left picture, which is the real thing, to our minimal version of it on the right, we are still very far from, the beautiful spindles that you find in real life. But I think it's important to emphasize that it's not per se the goal to reconstitute the complete cell or a cell module like a mitotic spindle in all its complexity. It's really about making a minimal version that allows us to understand the basic principles by which these systems operate, that we can control them and then integrate with other basic properties to make a really minimal version of a cell that can do all the things that I described in the beginning. So it doesn't have to be as precise, as sophisticated, as multitasking, if you want, as a real cell, but it's really about what is minimally required, which is very different from the really evolvable system that you will find in real cells. Okay, so in the last few minutes, let me go back to the bigger picture. So this was just a quick example from our own work. And this now, as I said, has to be integrated. So the collaboration is really meant to, not to spend another 10 years making each of our own experiments even more sophisticated, but really think of how can we combine spatial organization with DNA replication. Can we make a cytoskeletal system in a vesicle, in a liposome, where we are also producing the proteins from DNA that are needed to make this system? A little bit like Hannes was showing in the beginning, where he could produce the proteins that would then also be responsible for replication. So it's about integration, trying to combine this. And so we have a group of students across 6 universities and institutes that we really about brainstorm of how to do make bottom-up systems that can do more than one thing, and how do you have these systems talk to each other. And it could be that some of the ideas that we have for particular functions work very well in a single module environment, but are not very good for integrating with other functions, and that may change the choices we make for the design in the end of our synthetic cell. One maybe detail, or not a detail, is that all our work, for example, has been based on a eukaryotic cytoskeleton, and we probably will switch to more bacterial-like cytoskeletal systems for this synthetic cell project, because it's simpler and probably has a better chance of being integratable with other systems. I think it's for our consortium, and again, these are just my colleagues. I will not introduce all of them, but that apart from all these modules that we're trying to integrate, we need to think top down a little bit as well. So we have a few people also with expertise in whole genome optimization who take pieces of our modules and integrate them in minimal cells, the top-down way to see if they can be functional. John van der Oost is one of them, who is one of our CRISPR-Cas specialists. And then very importantly and interestingly also, we have a philosophist involved, because as was already mentioned, the question, what is life, of course, has been asked for centuries even. And I think during our program, it will be very interesting to see, at what point in time can we claim that we've created something lifelike or not? And what would the philosophers or the ethicists also, for that matter, think about it? Last few slides. So is this, I think, I really am convinced that there's many ways that this can be achieved, and we should try many ways and communicate about it between the different programs in Europe, but also the Build a Cell community in the US. So we are starting to get together more, also with industrial partners, because while I've emphasized the fundamental interest in, how can we use this bottom-up approach to understand life better, or at least cellular life, of course you can think of all kinds of systems that will come out halfway that can be really interesting for industrial applications. So maybe smart systems for drug delivery, or systems, cell-like systems that are optimized maybe to not grow and divide, but to produce a certain compound. And so there's all kinds of opportunities in different application areas where we talk also to industrial partners who may be interested in the final synthetic cell, but maybe are even more interested in the sort of the half products that we will generate along the way. So I think my time is up. I want to, of course, thank the funding agencies that throughout the years support this research, ERC being an extremely important part of that. All the many collaborators on the past work. It's really important, not for the bigger project, but even for our own work, to collaborate with different disciplines. Thank the people in my lab, of course, the most important ones of all. And this is them walking on the bottom of the sea in the Netherlands. And thank you for your attention.
So thank you very much, Marilène. That was a very nice talk. So now we're coming to the question and answer session. So for those people who are in the audience, you can click on the little icon and you can type in any questions that you would like to ask the speakers. I think I'll just go for a first question, which is of interest to me. And what I'd like to ask the speakers, what you think is, do you consider life to be really as simple as just assembling material building blocks?
Well, yeah, maybe I can start. So, what I consider life, I think I'm just not even going to pretend to have the answer to it, especially after the way Hannes introduced it. That's a very deep question. I think for our project, we decided to be very pragmatic and to say, okay, to understand life better, there's a list of things that we at least would like to understand better, and this is how metabolism, DNA, or information transfer, and cell growth and division can be coordinated as a way, already complex enough and difficult enough way to contribute to a better understanding of how cells work. And that's, is this life? Maybe, yeah. It's certainly, I think one important aspect that in the end has to be included is to understand also what design do you need to have a system that's also able to adapt and evolve, because that's one of the really important aspects of life that we don't impose from the beginning, but in the end I think will have to be an important part that needs to be added.
Yeah, I think I agreed with what Marilène said. So for me, a very important feature of life is that it can survive over time for many millions of years and adapt, even though the individual fragments or components are very fragile and have a very short half-life. So it's if you want, it's a dissipative structure, if you may wish, that has this ability. And I think the fact that it can adapt to a new environment is quite an important one in order for it to really remain functional in a changing environment. Because if we had no evolution, there would be no life on Earth anymore, because the moment you have an ice age or a climate change or whatever, then it will disappear again. So this is for me a very important feature, but of course it's not the only one. And as Marilène said, I'm also not in the position of finding now the definition, because there's so many people that think about it, very smart people. But I have, for me, of course, what's a very important feature for me is the aspect of reproduction and adaptation to a new environment.
Yeah, so coming after these two is very difficult for me now, but I think is what I'm thinking a lot about is what information is, yeah, because I think information content and how information can be stored and then changed and then reacts to the environments is, of course, also encoded in the central dogma, where you have a coupling between the RNA, DNA, protein information flows. So I think in a way, on a very abstract level for me, I fully agree that you probably need compartments to make life. Yeah, you need to have some kind of spatial entity, because all the life that we know is somehow compartmentalized. and you need some kind of information system, yeah, that transfers information from one generation to the next. So I think for me, on a very abstract level, of course, I realize the problem there is, of course, you have computer viruses that maybe, or computer systems that maybe fulfill the system, and I don't think they are alive. Yeah, but fundamentally, I think this aspect of information and retaining information and updating information is extremely important in these kind of systems, and the use of energy, of course.
If I may add, so even though I said that reproduction is very important, but again, it's one of these loopholes where you could say a mule, it cannot reproduce, but we would still classify it as alive. So this is one of the difficulties, that whenever you think you're close to a definition, that you find an example which then contradicts what you were just saying. But I agree with Ulrich that I think information is one of the main aspects. And but in the end, I think that there's no magic to it. I think in the end it all boils down to information theory, chemistry as a way of realizing it. So in principle, I think it should be possible to build such a system. Otherwise, I wouldn't be doing it.
We have a question probably for Ulrich. It is about the membrane proteins. So do you see a possibility that the cell would actually be able to fabricate the man-designed nanopores so that it would become part of the cell's own nature rather than something that you need to provide for each cell generation?
The simple answer is yes, because other people already have bacterial cells that actually produce these DNA origami structures themselves. So I think this is why I find this exciting, because in principle, if you can build everything from DNA and have some kind of templated assembly process, I think then only on DNA running, and there are DNA-based machines that also walk like the walkers that Marilene showed, but they are much slower. Yeah, so this life will be very slow on the slow lane. But fundamentally, I think the idea that there's some kind of, and the RNA world hypothesis that Hannes Mutschler also mentioned, that maybe everything started with RNA molecules and then moved to DNA, is also giving an idea that maybe everything can be done with one material system, but you have advantages if you add more complex systems. So, but the simple answer is yes, I think that you could make DNA, these DNA channels with DNA-based artificial cells.
So Ulrich, just to follow up on that. So you mentioned that you thought you could make a cell that was just made up of DNA and lipids. So how would you replicate your DNA in that scenario? You would also make, I don't know, DNA Lego enzymes that would do that?
So I think, so we in Cambridge here have people who look at self-replicating DNA molecules, and RNA is known to be also have more. So RNA is maybe the better alternative for that, but working with RNA is more difficult than with DNA for us, because it's also much more expensive. But fundamentally, there are very slow enzymatic processes also with DNA, yeah, where you can think about a self-replicating process where DNA doesn't make the DNA molecule, the building blocks, but actually if you provide them as from the outside, yeah, that then maybe you have a templated assembly of these kind of DNA molecules that will be extremely slow, of course. Yeah, but this is some of the ideas that you can think about, that you provide the building blocks and then things assemble into this structure. Of course, you need to provide energy, and I think this is one of the key things that you need to always provide energy, yeah, from outside.
We have a question from the audience about the DNA porins channels. The DNA itself is negatively charged. So how does this affect the molecular ionic flow through the pores?
Oh, okay. So this is actually good news that the DNA is charged, because it actually selects for positively charged ions. So we have a ion selective channel built. Yeah. So if you now want to have negatively charged ion channels, of course, with DNA, this will not work. Yeah. So we need to do then some chemistry, but there are tricks how to reverse the charge of DNA molecules. So you can make peptide nucleic acids where you change the backbone chemistry, and then it can be uncharged or positively charged even. So there are ways, but then of course you go into the realm of where you need to have more larger alphabet. Yeah, so like in proteins, we have 20 amino acids, and the reason for that is that you have much more functionality there. Yeah, and then this, if you want to have the same with DNA, you probably need to have more building blocks. Yeah, so more chemistry, and the cholesterol that we add is actually the same.
Okay, do we have more questions from the audience?
I don't see any at this point, but I think you had a question that you wanted to ask about life from earlier.
Well, the question is about whether you see that you are creating new kind of life, or maybe life with slightly modified properties, and maybe sort of half biological, half man-made life, because if you need to provide elements to very biological cells that don't arise biologically. So what sort of properties or nature of the cells do you might expect? So I can see a tiny twist of some properties of the cells, but do you have in mind something that would be quantitatively different from the cells that we currently have?
I can start if you like. Yeah, so I think so. It depends a little bit in the I'm sure this will go into different directions. So what we are now looking for is to learn how time and space molecules need to work together to get biological function. And so to do that, we define what we would like the system to be able to do, and we don't plan to replicate an existing cell exactly. So we're not even requiring molecules to come from the same species, or to use strategies for certain biological functions all coming from one example. So we use bacteria or yeast cells, simple cells, as inspiration source, but it's about the function in the end. So I don't think, and people tell us also, why rebuild an existing cell since we already have those cells. So it's not about rebuilding exactly the biological cells as we have them. It's more cell-like systems that can teach us about how real cells work, it might be a flight simulator or something you could call it. It's so simpler, naive, maybe not as capable, but really as a route towards better fundamental understanding. And that can be a mix and match of different real cells. If you think about applications, and I'm really convinced that if you get inspired by how biology makes things, or is sustainable in the way it takes energy and puts it into all kinds of functions, then if it's really to do something with these cells longer term, and that it can be simple things early on and maybe more complex things later on, then I can see that it will even be further away from real cells. Because then if you are really interested in making a cell-like system, a container that can make a product for you, or can pick up a signal in the body and then release something, or replicate itself to have many of these containers wherever it's needed in a medical application, for example, then nothing keeps us from mixing also with non-biological systems, as long as it has the function. So learning from the real cells, I think, will help us a lot also in making cell-like, life-like systems that can do things for us. And there, it's maybe even better if it's not totally biology-based, because then it can be more orthogonal, for example, to a human body. So there's not one answer to this question, I think. And for sure, we will not build exactly cells that already exist, because they anyway already exist. So we don't need us to do that.
Could I ask a question? So say you made some kind of autonomously replicating cell-like structure, and this somehow then was also released into the environment. Could you imagine a situation in which this would be able to will actually compete with the other living organisms that we have in the world around us? Could we end up in a situation where we had some kind of species that I don't know, maybe species is not the right word here, but something that doesn't usually live around us that could cause problems for the other things that do live around us?
So, I mean, I might start, but I think sort of systems, at least in the bottom-up approach we're building, they have, from my point of view, would stand no chance against anything that's out there, because they're way too fabricated, way too dependent, way too simple. And we can't even keep them alive, if you wish, in a test tube. So I think they would have a hard time to go into the wastewater and then do anything useful there. But that's my personal opinion. So I think we don't need to worry from that aspect. I think what is maybe a bit more critical is what has also been around before, that's genetically modified organisms. So things that, people are like, science is already doing that since many decades, that we alter already existing bacteria, for example, so that they can do new things. And of course, they are much more prone to escape and live outside. But then again, we have a lot of safety protocols that should prevent that from happening. And to the best of my knowledge, it has not really happened so far that a very hostile genetic modified organism is out there in the wild and does something horrible, even though you have a lot of antibiotic resistance genes that might be derived from the lab maybe, but then again, those things also emerge naturally. So, things are much better at evolving properties that they need already in the environment where they are, and they don't need us to implement them. But of course, we need to be aware of certain things, in particular with respect, for example, to CRISPR technology, where you can also alter higher order organisms or more complex things. But that's just my 50 cents. I don't know. I'm curious also on how the other two speakers see.
Yeah, I think I agree. I mean, compared to genetically modified real cells, these are way behind in their ability. But if we one day get sophisticated enough that they could do things that real cells can do, then of course we will benefit from all the the precaution that can be taken for genetically modified cells, which is sort of ahead in time for these kind of questions. So it's something to keep an eye on always. Of course, if you control bottom up, you can also build in an off switch, and you can make it dependent on crazy things that are hardly available. So it's more easy to make it, also to keep it weak in terms of being able to compete with other systems. It's also true, I think, I was talking about application possibilities, that making materials can also already be done with genetically modified existing cells. So we also don't need synthetic cells to do that. We already know how to do it. It will just be an alternative way that can be maybe more specialized, simpler to do one thing, and therefore be also more fragile. So that's, I think, in this safety discussion, a good thing.
Would be actually a safer option than GMOs. Right?
Yeah.
Okay, so maybe just because we're in the middle of a pandemic right now, I can just ask the question that people often ask about viruses. So, as Hannes pointed out, they're sort of simple organisms, but they cause a lot of havoc. So, are they really alive?
From my point of view, they are not alive as long as they're outside a cell. And the moment they enter a cell, and then, it depends on the definition. But if you say like metabolism is an important feature, they can't have it by themselves. But the moment they enter a cell and reprogram it, you might see like the infected cell as the virus being alive. This is one aspect. Other people might have different opinions, but the moment they're not inside a cell, I think they're more like a container for genetic material there to reprogram a host. Yeah.
Yes, I think I would say that they just contain information that can change a network in a running host. Yes, so in that sense, they should not be alive because they don't replicate themselves.
Okay, so then maybe on that note, I see that we've just run out of time. I would like to thank you all very much for taking part, and I hope that the audience learned something new. And so thank you very, very much.